Tihei Māori ora, he mihi ana ngā mana whenua ki tēnei takiwa, ka te māmoe waitaha, kai tahu whānui katoa, tēnā rā koutou katoa. E nā mana e nā reo raurangatirama tēnā koutou, kia ora koutou katoa. Ko Grant Robertson takawingoa, ko ngā tumu whakarai ki o tākou whakaihu waka, nau mai, haere mai ki tēnei whakanui. Greetings everybody, my name is Grant Robertson, I'm the Vice-Chancellor of the University of Otago, Otago Whakaihu Waka. Welcome along to this exciting event tonight, the inaugural Professorial Lecture of Beulah Leitch. I want to welcome especially all of you who are in the room today, friends and family of Beulah. I also want to acknowledge the extraordinary turnout from the Knox Church whānau once again for the second time this week. You are loyal supporters of your people, but also of us at the University of Otago. So thank you so much. And for me, lovely to see so many familiar faces in the room tonight as well. I want to acknowledge Beulah and your family, your husband Nigel, daughter Bethan and Sam, and Sam's parents, who are here tonight. It is terrific to see you all here. Anybody who works at the level of a professor in a stressful and difficult role with a depth of research relies on their family, but there are also enormous sacrifices that are made by family. And I think it's important on an occasion like tonight to acknowledge those sacrifices that you have all made to support Beulah. And I'd like us to show our appreciation to the family. I also want to make acknowledgement to all of Beulah's family and friends online who are joining us, particularly those who've gotten up in the middle of the night in the UK and Ireland to support her. Nau mai, haere mai, welcome along, and I hope you enjoy this evening, or this morning, as the case may be, lecture. Just before I say a couple more words about Beulah and hand over to Associate Professor Megan Gibbons, the Pro Vice-Chancellor of Health Sciences, I did want to make an acknowledgement that tonight's the final inaugural professorial lecture, ending on a bang, Beulah. But it's an enormous piece of work, and I just want to acknowledge the events team who organised this. And throughout the year, the lectures are an amazing part of what happens in our university, and it's a big effort. So given that it is the last of the year tonight, I do want to acknowledge the events team for their work. As I say, Megan will shortly introduce Beulah. Beulah will then give her IPL, and then Professor Greg Anderson from the Anatomy Department will finish up events with a thank you at the end of that. Beulah, from what I know, the depth of your research work is matched only by the support that you give to those around you, the manaakitanga that you provide for students. For those that you supervise, it expresses to me the whole person that you are. These occasions and gaining professorial status are about research, they're about teaching, and they're also about service. And I want to acknowledge all three of those as part of what you have done in your time with the University of Otago. I think it's 10-year anniversary next year from your arrival, 20-year anniversary, sorry, from your arrival. Time flies when you're having fun. And so that work over that 20 years is a work of extreme dedication. It's built off your strong reputation, which is international. And your ability to draw all of that together, and as the title of your IPL indicates, the making of connections is what really matters in making sure that we thrive as a university. You've done that, and we are extremely grateful for the contribution that you've made to us, and we are genuinely looking forward to this evening. But to provide a bit more of an introduction, I'm now going to ask Pro Vice-Chancellor Megan Gibbons to come forward. Nō reira, tēnā koutou, kia ora koutou katoa.
Tēnā koutou, rā rekatira mā e ka tini ka mahi mā o Aotakou Whakaihu Waka, tēnā koutou. E te tini e te mano o te hāpore, nau mai, ki te whānau, he mihi mahana kia koutou, nau mai, haere mai, e tēnei pau whakanui, Tēnā koutou, tēnā koutou, tēnā koutou katoa. It gives me great pleasure to introduce Beulah tonight, and it should have been Professor Lisa Matasu-Smith, but she is very unwell, so I'm stepping in her place. Normally I just finish up for the night, so it's nice to be at the front end. So let me start with Professor Beulah Leitch. She's an internationally recognised neuroscience researching brain synapses in health and disease. She began her academic career at Queen's University, Belfast, Northern Ireland, completing her BSc Honours in 1980, followed by an MSc at the University of Wales in 1981, and her PhD also from the University of Wales in 1986. After receiving her PhD, she spent a year as the head of the microscopy section, Department of Pathology, Midland Centre for Neuroscience and Neurology Hospital, Smethwick, Birmingham, the UK. Then from 1987 to 1989, she was a postdoctoral researcher at the University of St Andrews in the Department of Biology and Preclinical Medicine in St Andrews, Scotland. And from 1989 to 1993, a postdoctoral research fellow at the University of Cambridge in the Department of Zoology. From 1993 to 1995, Beulah was a lecturer in Science University of Durham in the School of Education, the Science Division, in the UK, and an honorary lecturer in Biological and Biomedical Sciences. In 1995, she was appointed lecturer in Biomedical Science at the University of Durham in the School of Biological and Biomedical Sciences. In 2005, Beulah joined us here at Otago as a senior lecturer in what was then the Department of Anatomy and Structural Biology, and now it's just Anatomy, and in 2016 she was promoted to Associate Professor. We're here tonight to celebrate her 2024 promotion to Full Professor. Beulah's research interests are in cellular and molecular neuroscience with a focus on brain synapses and their dysfunction and brain disorders. Synaptic dysfunction underlies a number of brain disorders, including epilepsy, ataxia, schizophrenia, autism, dementia, and Alzheimer's disease. She's particularly interested in changes that occur at synapses during neurodevelopment as a result of genetic mutations. One major research focus of Beulah's lab group at Otago has been investigating the underlying cellular and molecular mechanisms causing epileptic seizure in childhood absent epilepsy, which is genetic in origin. Another interest in new protein synthesis at synapses during learning and memory. Here the focus is on how altered synaptic protein synthesis may contribute to synaptic dysfunction and cognitive deficits underlying ageing and degenerative brain disorders. Her research has been supported by external grants and scholarships from the Royal Society, Neurological Foundation, Health Lottery Research, the German DAAD, OSTI Japan, Wellcome Trust the UK, Burroughs Wellcome USA, Biotechnology and Biological Sciences Research Council, and the Science and Engineering Research Council. Beulah has an international reputation for using cutting-edge, high-resolution imaging techniques for the detection of proteins at synapses. She's been invited as a research scientist to several distinguished, world-renowned neuroscience institutes, including the Californian Institute of Technology and the Okinawa Institute of Science and Technology in Japan, and the Max Planck Institute for Brain Research in Germany. She's published 55 full-length refereed journal articles and 3 book chapters, including in 2019 co-authoring with her collaborators at Max Planck a highly cited paper in the prestigious journal Science. Beulah is a dedicated teacher and supervisor. Since coming to Otago in 2005, she's taught science and medical students across all levels, from first-year health sciences, second-year dentistry, third-year medicine, and 200 to 400 level anatomy and neuroscience papers. Over the last 10 years, Beulah has supervised numerous postgrad students, including 4 MSc and 6 PhD students to completion. In addition to undertaking teaching administration and filling other department roles in the Department of Anatomy, Beulah was the director of the Neuroscience Teaching Program, one of the largest single major subjects in the University of Otago from 2021 to 2024. She was also chair of the Neuroscience Research Group in Anatomy, comprising 13 laboratories from 2014 to 2020. As noted earlier, Beulah's research interests are in cellular and molecular neuroscience with a focus on brain synapses and their dysfunction and brain disorders. Tonight, in her very appropriately titled talk, Beulah will be acknowledging the people and places, both personally and professionally, that have been important to her on her journey. The connections refer not only to people and places, but to one of the main focuses of her neuroscience research over the last 30 years, namely the connections between brain cells called synapses. Please join me in welcoming Professor Beulah Leitch to her inaugural professorial lecture, People, Places and Connection, the Importance of Networks and Connections for a Healthy Brain.
Thank you, Megan. Thank you, Vice-Chancellor and Pro-Vice-Chancellor, for your kind words of welcome and introduction. I think you've best given my talk for me. I also want to thank everyone in the audience for attending my IPL and those joining online, especially families sitting up for this. I'm really honoured and humbled and delighted that you're here with me on this occasion. And a very special thank you to my daughter Bethan and her husband Sam, and Sam's parents who've flown down from Auckland to be here tonight. Now mai, Harry mai, Céad Míle Fáilte, 100,000 welcomes in Irish to you all. You can tell from the accent I'm Irish. I want to begin my IPL by acknowledging and honouring my parents. Their love and support were instrumental in getting me to this point, where I am today. My mother died last year, and my father a few years before her from Alzheimer's disease. They were always my champions, encouraging and supporting me in all my academic pursuits and interests. This inaugural professorial lecture is dedicated to them. I also want to acknowledge the love and support of my other family members back in Ireland and in the UK. My sister June, brother Aaron, and my nieces Rachel and Miriam, and Miriam's two little boys, my great nephews, Ezra and Amos, and also my brother's daughters, who are all watching online with me. So, I think that already you've got the gist of my talk, but I'll still run through what I'd prepared. The title of my talk is People, Places and Connections, the Importance of Networks and Contacts for a Healthy Brain. I've chosen this title because in the IPL, as Megan has already said, I want to acknowledge who has been important to me in my life in getting to this position. So I'll be telling you about the people and places in the first half of the talk, and then I'll be telling you in more detail about connections, because connections refers not just to people and places, but to the synapses, the connections in the brain that I've been studying over the last 30 years. As Megan has said, my research interests are in cellular and molecular neuroscience, specifically the structural and functional development of brain synapses and their dysfunction in brain disorders. So I'll be explaining to you the development of brain synapses and their dysfunction in a specific brain disorder. As is traditional in IPLs, I'll be taking you on a journey of my career my pathway to this point. But first, I need to introduce you to the world of neuroscience. Neuroscience is the study of the whole nervous system with a primary focus on the brain. Your brain is amazing. It has over 86 billion nerve cells called neurons. To give you an analogy, that's equivalent to the 100 billion stars in the Milky Way, roughly. Brain cells talk to each other at points of contact called synapses. There are over 100 trillion synapses in your brain. A synaptic contact actually has a gap of about 20 to 30 nanometers. Now, just to give you a sort of scope on that, if you were to take one hair from your head, for those of you who have them, that would be 2,000 times thinner than a So that size, that gap, is 2,000 times thinner than a single hair from your head. So to see the gap, you need an electron microscope. This is what a synapse looks like in the microscope. It's taken from our work, and it's magnified 100,000 times. So what you're seeing in this is, just let me get my pointer up for you. This is the presynaptic site, and these are little vesicles. And these vesicles contain chemical called neurotransmitters. So the neurotransmitter inside those vesicles can either be excitatory like glutamate or inhibitory like GABA. And when the chemical is released into the gap, it binds to receptor proteins on the other side and allows the cell to communicate at these chemical synapses. So on this schematic, you can see a schematic of a neuron. It has a cell body called a soma. It's got the nucleus with the DNA, the instructions for making proteins in the cell, and it's got a long axon, and down that axon an electrical signal travels, and it causes chemical in the presynaptic terminals to be released into the gap. That binds onto the receptors on these dendrites, which are branch-like processes that come off the cell body, and then it causes the signal to travel down the next neuron. So you have an electrical signal turning into a chemical signal into an electrical signal. And so the nerve impulse is propagated from neuron to neuron. Each neuron is actually connected to many other neurons, forming these complex networks. These networks can comprise hundreds, even thousands of neurons, and they communicate with each other at incredible speed. Neurons transmit information to other neurons and to muscles and tissues throughout the body. That allows you to think, move, feel, and comprehend the world around you. This image is taken from a brain mapping project, and what you're seeing on it is stained in pseudocolors turquoise, a single neuron, and it's being connected to by 4,000 different axons. So this is part of a 10-year project by Google and Jeff Lichtman's lab to map the brain. And you can see from the complexity of that there's lots of interactions between different neurons in the cells, in the brain. So understanding how each of these nerve cells is connected to others is important in answering fundamental questions like how are memories made and what goes wrong in neurological diseases. That's what I'm interested in as a neuroscientist. However, I did not begin my academic career as a neuroscientist. So how did I get here? Okay. I was born in Belfast, County Antrim, Northern Ireland. Northern Ireland's colour-coded on this as yellow, and you can see that Northern Ireland is part of the United Kingdom. And I was born in Belfast, and the Antrim coast is famous for things like the Giant's Causeway. My father was from Belfast in Northern Ireland, and my mother from the south of Ireland, from Monaghan, and they met and married in Dublin. When they were first married, they lived in Belfast next to the Botanical Gardens. My father worked in the chemistry department in Queen's University on the other side of the park. June, my sister, was born first, and then I followed 2 years later. And then 5 years after me, my brother Aaron. And when, by this time, my parents had moved southwest from Belfast, about 10 miles, to a place called Lisburn. I attended Friends School Lisburn, which was a Quaker school, and is a Quaker school. It ranks amongst the top grammar schools in Northern Ireland. Entrance into grammar school at this time was based on an examination taken at age 11. So I studied all 3 sciences at school, and then my final year was a prefect and then deputy head girl. I went on to study zoology at Queen's University, Belfast, and my honours project was in parasitology with Professor Dave Halton, who was a truly inspirational teacher. He fired my passion in parasitology, and I graduated from Queen's University, Belfast with a BSc honours in zoology. And at that point, I was strongly encouraged to consider doing a PhD. However, I didn't want to continue a further 3 years in a research programme. My plan was to undertake a 1-year postgraduate course in parasitology with the intention of eventually working overseas with a development agency in Africa. That's what I wanted to do. So I applied for and was awarded a scholarship from the Department of Education Northern Ireland to study for a 1-year master's degree in parasitology. The scholarship allowed me the freedom to choose where to study for my master's degree. including the London and Liverpool School of Tropical Medicine in England. But I ultimately chose to go to Bangor University in North Wales. They also offered a 1-year postgraduate master's in parasitology. And my decision to go there was influenced by how beautiful North Wales is. Places mattered to me, and they still do, as you'll see throughout this talk. I didn't want to live and study in a large city like London or Liverpool. Bangor is a small cathedral city located between the mountains of Snowdonia National Park and the Anglesey Heritage Coastline. So after my master's, I again was encouraged by my lecturers that I should stay on at Bangor and do a PhD. They wanted me to stay there and said that it would be better for me to do that before embarking on any overseas work in Africa. So I wrote a PhD research proposal to work in Alan Probert's lab and applied for a personal scholarship from the Department of Education, Northern Ireland. My PhD was on the human parasite Schistosoma hematobium. This is a blood fluke that lives in the blood vessels around the bladder, and it causes the disease bilharzia. Schistosomiasis affects about 240 million people worldwide. It is ranked second only to malaria amongst the parasitic diseases with the greatest socio-economic impact. My PhD research was using ultrastructural techniques to study these parasites and their pathology and the effects of drug treatment. It was the skills that I developed in electron microscopy techniques during my PhD that were to prove important to my future career. I was awarded my PhD in parasitology and then planned to take a break and travel in Europe while deciding on my next career step. I was now having doubts about working overseas in a developing country. It was at this stage that I was called to interview for the position of electron microscopist heading the EM unit in the pathology department of the Midlands Centre for Neurosurgery and Neurology in Birmingham. So I decided to accept the position while I was deciding on which career direction to pursue. And it turned out to be a life-changing decision for me. It was at these meetings that I met Professor Edward Hitchcock. It gave me wonderful opportunities, this position, because I was presenting ultrastructural findings on selected cases at clinical meetings alongside the pathologist. And at these meetings, I met Professor Edward Hitchcock, a consultant neurosurgeon who was a pioneer in stereotactic techniques for brain surgery and functional neurosurgery. He invited me to watch him and observe him during stereotactic operations on patients, and also when brain biopsies were being taken for specialist EM analysis by me. So this launched me in a completely new direction. It marked the start of my career in neuroscience. I decided I wanted to undertake neuroscience research, but not in Birmingham. I said I disliked living in a large city in the English Midlands, far from the sea, and I'm aware that my in-laws are from Birmingham. So I resigned from the position and accepted a postdoctoral research position in neuroscience at St Andrews University, Scotland. I absolutely loved living in St Andrews. Working and living by the sea, I learned to scuba dive when I was there and went horse riding on the nearby beaches, and we had great Scottish ceilidhs. I was working on a joint collaborative project between 3 principal investigators, Bob Pittman, Jim Cobb, and Bill Heitler. And my postdoctoral research was into structural and functional development of electrical synapses. Now, these are also called gap junctions because they occur in other tissues as well. The gap at the junction, as you can see in this schematic, is much narrower than the gap that I previously described to you in chemical synapses. It's bridged by these thin tubes that allow ions to pass directly between cells. So at electrical synapses, you've got very rapid exchange of ions and very rapid propagation of an electrical signal. And this is what it looks like in the EM. You can see 2 sides of the membrane, and you can see the channels across it. Electrical synapses were first discovered in crayfish because they allow for that very fast flip tail escape response. and they've been studied very widely. So I published 5 papers from my postdoctoral research on these electrical synapses, and while I was in St Andrews, I attended my first Neuroscience International Conference over in Toronto in Canada. After the conference, I went on a seminar speaking tour to universities on the west coast of America, including San Diego, Berkeley, and Davis, then back to the east coast to Columbia, Yale, and Harvard, before then returning to Toronto and back to St Andrews. In St Andrews, we were also collaborating with Professor John Pitts and Malcolm Finbow at the Beatson Institute for Cancer Research, which is a very famous institute. They were generating antibodies to label proteins at gap junctions. And that's important for all types of cells, including cancer cells. And we were collaborating with them to use their antibodies to label the proteins in electrical synapses at the EM level of resolution, which was technically very difficult, but we managed to do that on isolated gap junctions and publish the findings. I was then offered a position in the Beatson Institute. But at the same time, I was invited to interview at the University of Cambridge by Professor Malcolm Burrows, a Fellow of the Royal Society and Professor of Neuroscience. He offered me a position in his lab in the Department of Zoology, so I decided to go to Cambridge. In Malcolm's laboratory, we had freedom to work on our own projects. I could direct what I wanted to do myself, so long as we were using the locust nervous system to investigate neurobiological questions. Now, you might ask yourself, why locusts? Well, invertebrates like locusts are used in neurobiological research as they have relatively simple nervous systems, fewer neurons, larger axons that are easily impaled so that you can study structure and function and behavior in the one organism. And lots of the pioneers in neurobiology have used invertebrates' nervous systems. Malcolm was author of the book, The Neurobiology of the Insect Brain. At Cambridge, I researched GABAergic inhibition, and you'll see later in the talk GABAergic inhibition is important to my later work, and within neural networks in the locust, and the development of inhibitory synapses. I also was collaborating with Gilles Laurent, who was a postdoc in Malcolm's lab as well, and we undertook painstaking experiments back into the embryonic locust brain. I was also continuing my collaboration with Bob Pittman from St Andrews, looking at modulation of transmitter release from muscarinic synapses. And again, muscarinic synapses will become important to you in the later talk. We all worked extremely hard in those days. Sometimes we were in the lab through the night for timed experiments, but we also played hard. I remember very competitive team field games and playoffs between other labs and science park companies. And on one infamous occasion, many of us ended up in Addenbrooke's hospital with injuries after a particularly competitive game of rounders. And as we got in, they kept saying, "Are you from the rounders game?" And we said, "Yes." Anyway, it was while I was at Cambridge, I also made the first of multiple invited research visits to the prestigious Californian Institute of Technology, or Caltech, in Pasadena to engage in collaborative research projects there. I was hosted by Gilles Laurent, who had by now been appointed as a new professor there. After my postdoctoral research at Cambridge, the time came to set up my own lab. One of the lectureships I was offered at that time was at the University of Durham. I decided to accept the position at Durham University, where I became one of the founder members of the new BSc Honours Biomedical Degree programme at Queen's Campus. Accepting that lectureship at Durham University turned out to be one of my best career and personal decisions, as that is where I met my darling husband, Nigel, who was already an established academic there in the Department of Biological Sciences, teaching and researching in behavioural ecology and conservation and doing work with film companies, documentary wild films. So we were married in Durham, and Nigel planned a surprise month-long honeymoon in the Seychelles and then in Africa, including a horseback safari. So I got to see Africa with Nigel, and there's a whole series of other tales to tell with that regarding my level of prowess with riding and seeing that you had to gallop out of trouble. Anyway, I had doubts about whether I'd be able to do that, but we got back safely. And back in Durham, I was setting up new projects using rodent models to study changes in synaptic proteins at synapses underlying brain disorders. So that I had a number of projects underway, but I was also had ongoing projects on locusts with Gilles Laurent at Caltech. It was while I was on a research visit to Caltech that I suffered my first miscarriage, the first of 6 in total. I flew back to Durham and was admitted to hospital. After further miscarriages, Nigel and I moved to the countryside with our horses for a better work-life balance, following the advice of my consultant to reduce stress. And in the following year, which was the new millennium, a host of births occurred, including our darling baby daughter, Bethan. I wasn't allowed to work by my consultant throughout the pregnancies, and as soon as I was pregnant with Bethan, he stopped me from working, and I had a year's maternity leave. And after Bethan was born, I was off then for quite a while. So I effectively had 2 years break while the period that Bethan was born from academia. Fast forward 4 years. Bethan is now 4 years old and in her first year at Durham High School for Girls. I am back teaching and running my research lab at Durham University, but I'd now decided to stop travelling overseas on collaborative research visits while Bethan was young. Nigel was still continuing to do his research and teaching in the university and taking students on field expeditions over to South America and South Africa at this time. And then Nigel was offered a professorship in zoology at the University of West Indies in Trinidad, and also to set up a biodiversity institute in the Caribbean. We were all set to go, but at the last minute pulled the plug on it due to health concerns with Nigel. At around about this time, Nigel spotted a lectureship advertised at Otago University that was in my field. Nigel knew that I was interested in living and working in New Zealand. It had a reputation for being green and clean, which appealed to me. So he said if I wanted to apply, he would support me and back me. So I did apply and was offered the position. So then we had to decide, should we stay or should we go? By this time, we were ready for a new challenge, but it was still a difficult decision to give up 2 permanent academic positions at a prestigious university in the UK. The final decision to go was based on 2 main factors. First, the head of department at Durham, Brian Huntley, and the University of Durham offered to hold Nigel's position open for 3 years if we wanted to return. And secondly, we had a guaranteed place for Bethan at Columba College by the principal, Miss Wilson, at that time. And so we came to New Zealand. We arrived in New Zealand in August 2005. I was appointed as a senior lecturer in the Department of Anatomy and Structural Biology, as Megan said it was then called, by Professor Helen Nicholson, who was the head of department at that time. So now I want to change gear and tell you more about my research here at Otago. My research interests since coming to Otago have included investigating how changes in synaptic proteins resulting from genetic mutations cause functional abnormalities underlying brain disorders such as childhood absence epilepsy and associated comorbid behavioral and psychiatric disorders. Another focus of my interest has been into the synthesis of new proteins at synapses during synaptic plasticity. Synaptic plasticity is the ability of synapses to change, allowing them to adapt to new information, which is important for learning and memory. I'm interested in how altered local protein synthesis at synapses may contribute to synaptic dysfunction and cognitive deficits underlying degenerative brain disorders such as Alzheimer's disease. So now I want to tell you a little bit about one of those topics, childhood absence epilepsy, that we've been investigating. A major research focus of my lab has been on childhood absence epilepsy to investigate underlying cellular and molecular mechanisms causing seizures in these children. This is fundamental to identifying targets for future treatment strategies. Childhood absence epilepsy affects children around the age of 4 to 14 years old and accounts for 10 to 17% of all cases of epilepsy in school-aged children. Epilepsy is actually a group of diseases. It's not a single disorder. And it's characterized by epileptic seizures. So, although there are many different types of epilepsies, you can divide them into two main groups, focal and generalized. Focal epilepsies are those ones that originate from abnormal firing of brain circuits in one part of the brain, in one hemisphere. Generalized applies to both hemispheres. So in generalized, there are groups of neurons that are firing off in both sides, both hemispheres, and childhood absence epilepsy fits within the generalized category. So, to investigate the underlying mechanisms causing epileptic seizures in these children, we use a rodent model which has naturally occurring absence seizures. This is the stargazer mouse. The mouse has spontaneous absence seizures that appear at a developmental stage in the mouse that is equivalent to the age of their onset in children. So the key things that I want you to remember as we go forward in this lecture is these types of these types of generalized seizures affecting both sides of the brain in a child are genetic in origin. So they're genetic, they're generalized on both sides of the brain, and they're non-convulsive. They comprise just brief periods of a few seconds of unresponsiveness where the child might look like they're vacant, staring off into space. And often it's picked up at school, the child might be accused of daydreaming, not paying attention. So they're called absences. But the key features are they have a hallmark signature on an EEG, a brain recording, called a spike wave discharge, a spike and a wave. That's a classic feature that is for absence seizures, associated with behavioral arrest when the child starts moving. It's probably best illustrated if I show you a YouTube video of a little boy having an absence seizure that his father has posted on YouTube, but he's recorded it because clinicians often want to see a video of a child when the reported is having a seizure. You're going to see him blowing on a rag that induces an absence seizure, and it'll only last a few seconds, and then he will start blowing when he comes out of the seizure. But he doesn't hear his father calling him, and he's unaware of anything that's happening at the time. So he's blowing, and I'll tell you, he's about to go into the seizure now. Just stares blankly into space. He doesn't know what's going on.
Tyler.
So he's totally unaware. He doesn't hear his father calling his name. When he comes out, he starts blowing again. Now, that doesn't look particularly dramatic, and indeed, it used to be called petit mal seizures because it was less dramatic than the tonic-clonic seizures that people think about when they think of epilepsy seizures, where somebody has a convulsion, falls to the ground, and starts to jerk around. In absence seizures, the child will just be vacant like that for a short period of time. But that can happen hundreds of times a day, and we now know that these are not benign. There are a number of comorbidities associated with these seizures, and there are dangers that if a child was to have one of these seizures, if they're not accompanied, they could drown if they were in a swimming pool or if they were out cycling. And there are all sorts of other things going on with these seizures. So they're certainly not benign, even though they don't look dramatic. So what is the solution? Anti-seizure medications, as in all cases of epilepsy, are usually the first line of treatment. And for childhood absence epilepsy, one of the drugs that is the first line drug used, even after 50 years of being, since it was made, is ethosuximide. So this was produced 50 years ago. It's specific for childhood absence epilepsy, but as with all anti-epileptic drugs, it's only effective in about 70% of patients, and there are side effects. So that means there's a 30% failure rate, and they cannot cure epilepsy. They only aim to reduce the seizures. So there's a desperate need to have more targeted drugs that are more effective and specific for an individual patient, that are targeting their underlying mechanisms so that there are fewer side effects and they're more effective. And to develop such drugs, you need to know what's happening down at the cellular and molecular level to be able to identify targets that could be used for future drug treatments. So what I want to do is just show you in this slide what we do know from human and animal models. So in rodent models, just like in humans, they have the characteristic spike wave discharges with the behavioral arrest, okay? We also know from MRI, functional MRI studies in humans and also from tests in animals that these spike wave discharges on the EEG arise within circuits connecting the cortex to the thalamus. So you can see in this schematic where the thalamus is on it. So we know those factors, but what we don't know fully is what's going wrong within these circuits down at that cellular and molecular level that's responsible for causing the seizures. So that's what we've been interested in looking at, is what's going on at that level in our mouse model. So first of all, bear with me, I've got to introduce you to the CTC network. I promise this will be painless. So what I'm going to do is just show you a section, a schematic taken out of the brain at the level of the thalamus. So if I magnify this on the right side, this is grossly simplified, but what you can see here is a representation of the cortex, and this is the thalamus, which is beneath the cortex. And the thalamus is made up of many different parts, but one of the key parts is the dorsal thalamus, and then around the outside, there's another part of the thalamus called the RTN, reticular thalamic nucleus. All you need to know is that this has got a shell of inhibitory neurons in it. So the thalamus is the gateway for sensory information coming in from outside the brain. It comes in, and there are excitatory cells in the thalamus. They send a connection up to the cortex, excitatory, and the cortex sends an excitatory connection back down to them. The important thing is there are also these inhibitory interneurons that are in that shell of exclusively inhibitory interneurons around the outside of the thalamus, and they're putting on inhibition onto these excitatory neurons, and there's the equivalent up in the cortex, inhibitory interneurons there too. Bear with me. So I'm going to tell you a little bit more about those inhibitory neurons. Here they are up in the cortex. They've got GABA in them, an inhibitory transmitter. And down here in this shell around the thalamus, there are the inhibitory neurons again. And what they do is they're strategically placed so they can act almost like the brakes preventing runaway excitation. So when the excitatory cells in the cortex and in the thalamus are firing, to stop those overfiring, you have inhibition within the system. That's grossly simplified. But the key thing is, in the normal system, the way that these inhibitory and excitatory neurons interact with each other underlies a normal oscillatory rhythm within the thalamus, okay? That's processing sensory information coming out. And somehow that gets transformed into a pathological rhythm that is represented by that spike wave discharge. It means that there are neurons firing excessively together, causing this spike wave discharge that underlies absence seizures. And what we want to find out is, how is that happening? So to do that, we use a rodent model. This little mouse, the stargazer mouse, has the classic features of spike wave discharges, absence seizures. It stops walking when it's having them, so there's behavioral arrest. When it comes out of the seizures, it continues walking. And I said to you that these seizures are genetic in origin, so there's something gone wrong in the genes on it. So it's easier to look at this in a mouse model because it's got a single genetic mutation. In this little mouse model, the mutated gene encodes for proteins. Genes make proteins, and proteins carry out the functions in the cell. And the protein that's mutated in these little mice is called stargazing, after the little mouse. But it turns out that this protein is in fact one of a group of a family of proteins called TARPs. Now, before you start to go to sleep, you don't need to worry about that. All you need to know is that the job it does is almost like a little bus shuttle. It associates with receptors called AMPA receptors. AMPA receptors are receptors that respond to glutamate, and so they're responsible for the fast excitation in the nervous system. And what these little proteins do is they associate with the fully formed AMPA receptor, which is 4 proteins, and they shuttle them up into the position in the postsynaptic membrane where they can respond to glutamate and cause the neuron to fire, okay? So if you have a gene that's mutated and it's not properly producing a protein that's responsible for putting AMPA receptors into the synapse, you get AMPA receptor deficits. So what we wanted to do was to find out within the CTC network, where are there AMPA receptor deficits? Where could those be targeted, and do they actually cause seizures or something further down the track? So ultimately, we did a huge number of experiments at different levels, but we had to come down to the EM level so that we could look at AMPA receptors at the synapse in our epileptic little stargazer mouse and also non-epileptic littermates, because we wanted to see and do quantitative analysis to see the relative density of AMPA receptors both in the epileptic and the non-epileptic mouse and at which synapses those were occurring at. So this is what this is showing you. It's just a schematic of the fact that if you fix tissue for the EM, what you're doing is you're cross-linking proteins to get really lovely ultrastructural detail. But that destroys the proteins. So you have to do fancy EM, all different sorts of techniques that need to be adjusted for each protein, where you're doing minimal fixation. It's a bit like fixing the proteins is a bit like cooking an egg. You're coagulating the egg, and so that's no good when we want to label the proteins in it. So what we do is we do fixing that is very light using freeze substitution, all sorts of things. Don't worry about it. All you need to know is to identify these AMPA receptors, we take one antibody to a protein in the AMPA receptor, and we label it in our special tissue fixation method that doesn't destroy the protein. And then we take a second antibody to that first antibody, which has a gold probe on it. Now, the gold probe is electron dense, so you can see it in the EM. And that's what you're seeing here, are these dots, or the gold probe relatively labeling the AMPA receptor. So that's what we did. Two PhD students in my lab, Susanna Barrett and Olga Shitsova, were working on this project, and they were looking at AMPA receptors down in the thalamus. So what they did, they took hundreds and hundreds of sections and did labeling to get the relative quantity of gold labeling in non-epileptic littermates compared to the stargazer epileptic mice. And in a nutshell, there was a significant reduction of AMPA receptors in the epileptic mouse. But they were only occurring at synapses onto these inhibitory neurons, which is important. Nadia, another PhD in the lab, was working on a project up in the cortex, and she was looking to see whether or not AMPA receptors were also lost in the cortex on these inhibitory neurons. But this required a bit further step on it, because our inhibitory neurons weren't in a nice shell that we could take a plug out of the shell and know it was just the inhibitory neurons. These were mixed in with all sorts of other neurons, and so we had to use a second antibody against a protein inside these inhibitory interneurons, and that protein is parvalbumin. So that's just important to remember, that these inhibitory neurons, as well as having GABA neurotransmitter, which is inhibitory, they also have a protein called parvalbumin PV, and we could identify it with a larger gold probe. So anyway, the key feature of this is, again, there was a significant reduction in the epileptic mouse at AMPA receptors onto these inhibitory interneurons. So that meant that activating, putting on the brakes, there was nothing there to put on the brakes on these interneurons, both in the cortex and in the thalamus. And importantly, Nadia also went back and did this on post-embryonic stages, early stages, and we could show that this loss of AMPA receptors was happening just before the onset of seizures. So therefore, they could be causative of the seizures. That was important to establish because another PhD working in my lab, Steve Seo, he was working on GABA receptors, and we'd shown that there were alterations in GABA receptors, and this was published in the International League Against Epilepsy's own journal, very high-ranking, Epilepsia, and it featured on the front cover. And we were able to show that the changes in the GABA receptors were happening after the seizures. So they weren't causing them, but they were a downstream effect, probably compensatory. Anyway, to put that all together for you, in the stargazer, it has a genetic mutation that affects the protein stargazin, which is a TARP that shuttles these AMPA receptors into the PV interneurons. And the PV interneurons put on the brakes, put on feed-forward inhibition. So if you don't have AMPA receptors on them, it's like cutting the brake cables. You can't slow down or stop the overexcitation in the system. So if we lose feed-forward inhibition, is that causing absence seizures in these mice? Well, But to prove that, we needed to do another series of experiments. And we asked the question is, if we silence these PV interneurons in normal mice, does it cause seizures? Because if it is causative, then if we turn them off, we should be able to see seizures. And to do that, we had to use more cutting-edge technology called DREAD technology. Now, I know you're starting to groan under the names I'm giving you, but bear with me, I'll try to make this simple. Designer receptors, DREAD stands for Designer Receptor Exclusively Activated by Designer Drug. And basically, these designer receptors, so the receptors in the synapse, have been engineered so that will no longer respond to any chemical, any neurotransmitter in the brain, but only to a synthetic drug, a designer drug, okay? So it's a bit like changing the locks in a door. You're changing the locks in your synapse. So what we do is we take a designer receptor, which is like our new lock, and when we apply our designer drug to it, we can control whether the cell fires or whether we silence it, okay? Now, you remember I talked about muscarinic receptors before? The actual designer receptor is a modified muscarinic receptor. All you need to know, don't worry about this, all you need to know is that we've got a receptor that naturally occurs that is responsive to acetylcholine, okay? So they're called G protein coupled receptors because the receptor has associated with it a G protein. And that G protein sets off a train of events that will cause a cell to fire or to be inhibited. If it's got a GI protein, then it decreases neuronal firing. If it's got a GQ protein, it increases neuronal firing, okay? So that's all you need to know. So we've got a GI DREAD, an engineered receptor that now will only respond to our designer drug, which is CNO. And if we apply CNO to the GI DREAD, we get neuronal silencing. If we apply it to a GQ DREAD, we get neuronal activation. So what we wanted to do was put our designer drug into our PV interneurons, apply our drug, and silence the PV interneuron in normal animals and see if we get seizures. Alternatively, we want to put in the excitatory DREAD so that we can activate the PV interneurons and in an animal that has seizures to see if we can control the seizures. So that's the theory of what we wanted to do. Of course, you have to get these into the animals, okay? So again, we used breeding, and all you need to know from this is that we have GI DREAD mice, shown here in this grey and green, and we have PV Cree mice. They're both transgenic mice. They don't have any epilepsy, but this mouse has the inhibitory DREAD already in its cells, but it's not activated. It's got the machinery to make it, but there's a stop signal. But if it is crossed with this transgenic mice, it's got like the molecular scissors that can cut out that stop signal, and it can do it just in PV cells or whatever Cree mouse it is. So effectively, we could use this to create GI DREAD mice that only had the designer receptor in the PV neurons, and then half the litter would be non-DREAD controls. So that's what Sandesh, another PhD in my lab, did. He came to work in this project with me, and he took the GI DREAD mice, and we recorded from them, just like we would record from a child's brain, and then video them to see what they were doing. We did recordings and video on mice moving around, and before CNO, this is just what the trace would look like. As soon as we applied the CNO, the designer drug, we were activating our inhibitory DREAD, so we were silencing the firing of these neurons in normal animals. And we got bursts of spike wave discharges, and the animals stopped moving around. And then when it came out of them, it moved again. So we had our classic features, spike wave discharges on the EEG and behavioral arrest. And you didn't get any effect if you just injected a solution in which the drug was dissolved. You had to have the CNO drug. And then Sandesh repeated this on non-DREAD controls, and there was no effect. So we had shown in this experiment that if we use DREAD technology to silence those PV interneurons, we get spike wave discharges and behavioral arrest, which are the hallmark features of absence seizures. And in the alternative experiment, where we induced absence seizures in animals and used an excitatory DREAD technology so that we could excite feed-forward inhibition during the seizure, that prevented and reduced the number of spike wave discharges. So, impaired feed-forward inhibition may be just one underlying mechanism for spike wave discharges in a population of patients with absence seizures. It's never as simple as that. In patients, they have many different genes that are affected. It's polygenic. So we have different models. This is just one model that's spontaneous seizures, so this might be one mechanism in patients who would have this underlying abnormality. What's key is that we need to discover the different types of changes at the genetic level, at the cellular and molecular level, that would be targets that could be used and directed new drugs towards. So now, breathe, this doesn't take too long. The next one I want to tell you about is just briefly some of our interests in local protein synthesis at synapses. In the next few slides, I'm just going to tell you about a collaboration with the Max Planck Institute for Brain Research in Frankfurt in Germany. Erin Schumann is the director there, and we have collaborated on this project. Now, previously I said to you, genes make proteins, and proteins carry out the function in the cell. And in most cells in the body, you have a nice cell and a cell body and a nucleus with the information to make the protein, and the protein can be used in that cell. In neurons, because of their long axons and dendrites, it's not so straightforward. Proteins might need to be transported right to the end of those axonal terminals to be able to do their job. And the challenge is how proteins can be supplied to remote regions where synapses are made with other neurons. Synapses need to rapidly change their proteins during learning and memory, so transport from the cell body might be just too slow in some cases. So one solution is to make them locally down at the synapse. And there's been a wealth of information to show that local protein synthesis occurs in the dendrites. Erin Schumann's lab and others have shown this for many years, but it's been very difficult to show that this is happening on the presynaptic site, and that's for a number of reasons to do with size and techniques and everything. Anyway, we managed to do that using our combined expertise. This was an international collaboration between scientists from the Max Planck Institute, Otago, and the Institute for Neuroscience in Bordeaux, 3 PIs and postdocs. And effectively, what we were able to do was to show that the presynaptic site of the neuron, both in excitatory presynaptic terminals and in inhibitory presynaptic terminals, they had the machinery to make proteins on the presynaptic site. And that, furthermore, the axonal terminals were able to make proteins that participate in synaptic transmission. So we used various adaptive metabolic labeling techniques, high-resolution imaging of proteins being made with these techniques at the electron microscope level and the expansion level. And this paper, the novel findings culminated in a highly cited publication in Science, one of the highest-ranking scientific journals in the field, and it received enough citations to place it in the top 1% in its field. So that concludes an overview of some of the important research. I haven't been able to cover a lot of the research that I've done here at Otago with other colleagues, but I want to just acknowledge the research funding for this work. We've been funded through agencies here in New Zealand, in Germany, in Europe, and also in America for the research I've told you. Of course, as Grant said, research is just one aspect of an academic's role. Teaching and service are also an important part of the job. I've taught anatomy students at all levels, through from first-year hubs right through to postgraduate and dental and medical students. And as Megan already said, I've been director of the neuroscience program from the beginning of 2021 to the beginning of 2024. And during the time that I've been director, the Otago Neuroscience Student Association, who are here tonight, was formed, and they've been very active in interacting with students and in outreach activities with the program, so we thank them. And also one of my service roles has been as chair of the Neuroscience Research Group in Anatomy. I was chair from 2014 to 2020, And the neuroscience research group in anatomy at that time comprised 13 different laboratories, and you can see on the right-hand side the PIs that are responsible for those laboratories. And we held regular retreats at St Margaret's College and had research speakers. But as part of that, I chaired meetings, and we had various members of the different labs, including from mine and other labs, who were going out into schools and doing research into schools and telling them about neuroscience, and also at events, at festivals, etc. And these groups were spearheaded by Nico Vutrale and Mick Watts, so I want to acknowledge them. And in this slide, which looks like a lot of names on it, on the left-hand side are some of the students from my lab, some of them having dinner at my house and things on it, and I was out for things. So I want to acknowledge that there were a lot of people involved in different levels of research that I haven't been able to tell you about on the research team. I also want to acknowledge that I've had a lot of help from my New Zealand co-authors and collaborators, and from collaborators overseas. And there's been help, obviously, and support through technical staff and support staff at all levels, from client services right the way through to technicians. And I had some superb mentors and advisors since coming to Otago. But I want to give a special shout out to Christy, Kate and Marcel. These are my WILO ladies. And WILO stands for Women in Leadership at Otago, and we were part of the women selected in 2018, and we were put into a team, and we still regularly meet, and they support me, and we give each other therapy. So what I would like to just finish off in the last two slides, you're glad it's the last two slides, a big thank you to my family. As always, my thoughts go back to my parents, who were amazing, my loving husband, my beautiful daughter, and Sam, the son I never had. He's a credit to his parents. And in the opening sequence, because I was getting tongue-tied, I should have mentioned my nieces, Nicola and Lauren, my brother's daughters, and I mentioned Rachel and Miriam's two little boys. And I loved my parents dearly and still do. And the very final slide, but not least, goes to my church family. It's been important to me throughout my career in life, all of my church connections and the people who have supported me. And there's been so many of them. But here at Knox Church, where Bethan and Sam were married, and in Highgate, we've had wonderful friends and supporters. And also Father Mark from the Catholic Church, who runs regular retreats, which have just kept me uplifted throughout my time at Otago. Thank you very much, and thank you to God. Amen.
Kia ora koutou katoa. Hiori ahau no kotiranga inga ranga miwera ingara I te pū ake ahau I o tūtahi. Ko Greg Anderson tōku inga. Which is to say that I'm Greg Anderson, I'm the head of department in the Department of Anatomy, and I come from some of the same places that have been part of your journey. A little bit further back, mine's 2 generations back, and that's why we sound different. So. Beulah, I want to thank you for a wonderful talk, and I'm going to come back to that. But firstly, also for all that you've been to our department over these last 2 decades. And your passion for neuroscience has been evident in all the aspects of what makes a professor, in your teaching, in your service, in your research, for example, through the neuroscience program, and just to mention one, and I think I can safely, on behalf of your postgraduate students, really extend a thanks for the mentorship that you've given. So many, many thanks. Well, the synapse, I mean, I think our eyes have been opened here, whether we're neuroscientists or people from some other skill set and background, as to this wonderful structure. And not to downplay the 100 billion nerve cells that are there, but they don't do that much across our lives. They don't change that much. It's those 100 trillion, was it, connections or synapses that make us the dynamic changes that we are as we learn, develop skills, and then potentially neurological diseases as well. So you've illuminated the synapse, and thank you for the wonderful imagery as well. Not necessarily in beautiful fluorescent colour, because the electron microscopy, it's not like that. Its claim to fame is the enormous zooming in to a structure that's millionths of a millimetre, the synapse. And every one of those dots, am I right in saying that was a single receptor? Is that quite right? Or a small group of cluster of receptors? I mean, the scale of the magnification that you image, well, you showed yourself in your opening slide, this is the biologist's equivalent of looking out into the universe, seeing things which can't be imagined with the naked eye. So thank you for illuminating that for us. We have a gift to give you. I'm told it's right here. So it's just a small token of our appreciation. So there you go, and I'll have to give you a hug one more time. For those of you who can, we would love you to continue to celebrate with us. We have some drinks and nibbles over at the staff club, which just follow the crowd, but it's one of the old brick buildings across this way, just 1 minute's walk. And so if you would like to join us there, that would be wonderful. Thank you once again, Beulah.